Hello, everyone, and welcome to Module 6. This is our first lecture about lipids, and we're first going to talk about classification and functions, and then we'll go into talking about digestion and absorption. So our objectives for this part of the lecture are to describe the characteristics and functions of lipids and list the different types of fats, including their clinical significance. Okay, so lipids are mostly composed of carbon and hydrogen, hardly any oxygen, and you can see that in these figures here that they're mostly carbon and hydrogen. And this chemical composition makes them highly hydrophobic and therefore insoluble in water. And because they have more hydrogens, if we think back to our other lectures, it's really this removal of hydrogens from our macronutrient that is going to ultimately end up with a majority of our ATP synthesis through the electron transport chain. So if you look at just the structure and see how many more hydrogens there are on this fat molecule compared to the carbohydrates that we just talked about, then you can understand that they are more energy dense. They have more energy per gram. And we definitely see that because we know that while protein and carbohydrates each have 4 calories per gram, that fat has 9. And also other dietary characteristics are that there are essential fatty acids that we must get from our diet, and we also need fats to promote absorption of fat-soluble vitamins. Okay, so this was the dietary guidelines for 2015 did have some changes in fat recommendations compared to prior guidelines. And we are in a few weeks on our last week that we'll be doing lipids. We're actually really going to look at the evidence behind these changes because this is really new for a lot of us in nutrition. But I can say about this dietary guidelines that the recommendations for fats are supposed to be those that are associated with the lowest risk of chronic disease while providing essential nutrients. And the final guidelines do say, continue to say that we should limit our saturated fats to about less than 10% of calories. But sorry for this little typo here, but there are new ideas coming out about saturated fats too, and that's what we're going to talk about in that week. Another thing about the saturated fats is that part of the recommendation to keep it to a low percent of calories is just that if we include too many, then we can't get all of the nutritious foods that we need and still fit within our caloric recommendations for the day. The guidelines do say to limit. I wonder if I have an old lecture here. To limit trans fats and cholesterol as much as possible, but the recommendations had prior given a specific recommendation of less than 300 milligrams of cholesterol, but in these new recommendations, I'm sorry, that's not really clear, but make a note of this. In these new recommendations, there's really not a hard number for cholesterol and where we should keep that. So for fat intake as part of percent calories, there are higher recommendations when for children and young adults, and then in adolescents, and then in adulthood, our AMDR is 20 to 35%. Okay, so fats have a lot of functions. So the first is that they are so efficient at storing energy. And part of the reason is the reason that we just talked about before, that you can get more energy per gram of the macro molecule. And this saves space basically in your body. So we have less oxidized material and more energy compared to glycogen, which we've talked about in the past. Lipids are also necessary to make other necessary lipids that we need for our biological processes, such as we can endogenously produce cholesterol. We need obviously phospholipids for our cell membranes and so on and so forth. And we're going to talk about everything really that's listed here. And we also need it for vitamin D production, which I'll leave that for micronutrients. Another function of fat is that it pads our bodies and our vital organs to help protect them against danger or impact, and helps us to maintain body temperature as it serves as insulation. And as we just said, they are also crucial parts of the cell membrane. Okay, so there is many different ways to classify fats. For the most part, we refer to fats by what's called their trivial name or their common names, and then there's also their scientific names. But really, there's so many different ways to group fats that this could be a whole lecture in and of itself. But I don't think this would help you as much in your profession. So I'm just going to go through the fats that are of the most significance in our body systems and those that we would utilize as a dietary source of energy. So first, this will be a few slides where we're going through just the major types of fats that are pertinent. So first is the non-esterified fatty acids. So this includes our saturated fatty acids and our unsaturated fatty acids. And as we all know, unsaturated fatty acids have a double bond between carbons so that they are not completely saturated with hydrogens, such as SFAs. We then have our glycerolipids, which is when these fatty acids from above are esterified onto a glycerol and become a tri-di or monoacylglycerol. So, we'll talk, of course, a lot about triacylglycerides or glycerides. And this is the same thing as a triglyceride. It's just kind of the more scientifically correct way to say it, but those can be used interchangeably. Okay, we then have our glycerolipids, which are I do want to take one step back, to number one, which is the trans fatty acids. So again, I think this is something that we all know, but nearly all fatty acids that exist in nature, when we have a double bond, the remaining hydrogens are on the same side. So this is a cis bond, but when the hydrogens are on the opposite side, it's a trans bond, and that's called the trans fatty acid. So we don't have many of those in nature, and those that are or those that are manufactured, such as those we find in margarine or shortening or some of our frying fats that are hydrogenated, do have these trans fats, and the dietary guidelines tell us to avoid trans fats as much as possible. Okay, then we have our glycerolipids, which is our phospholipids, our cardiolipids, platelet activating factor, and our lipoproteins, and we'll go into more detail about some of those. We have our sphingolipids, which I won't go into too much about, but you can find out about in your textbook. And these are crucial for various signaling functions. We then have our isoprenoids. And again, I won't go into this one as much, but they are compounds responsible for colors such as beta carotene, aroma, and chemical signaling in plants, and are also important in CoQ enzyme and the electron transport chain, and in squalene, which is a precursor for sterol synthesis, which we'll talk about next week. We also have our steroids, which we'll go into more detail on next week, and bile acids. We have waxes, which are less common, but they're often on they cover a plant. If you think about some of those plants with those waxy leaves, and it's also important in humans in the mode of vernix, which is that waxy white stuff that's covering an infant's skin in late gestation, but often when they're born too, you'll see this white stuff. And it's always been common practice to wash this vernix off, but now we're realizing that it is actually protective, especially could be beneficial because it protects against bacterial infection. So now we're moving toward recommendation where we actually leave vernix on until it is absorbed. But that's a little bit of a side shoot. So we won't talk much about waxes, but that is a major type of fat. And then we also have eicosanoids that are associated with our essential fatty acids, and we'll talk about those in detail in 2 weeks. Okay. Okay, so let's talk about fatty acids a little bit more. So we have we know that our fatty acid chain is nonpolar, and that free fatty acids are really not that ubiquitous in the body. Usually they are esterified to a glycerol backbone, especially in the form of triglycerides. They have this carboxylic acid head and this hydrocarbon chain tail. And we classify fats often according to their chain length. So the shorter, the chains with less than or equal to 6 carbons are short chains, and then our medium chain and our long chain fatty acids. So these, like, for instance, we'll hear about medium chain triglycerides in MNT treatment sometimes. Okay, so that was our fatty acids, or our fatty acetyl, and then we have our acylglycerols, where the fatty acids are esterified, which means they're connected to a glycerol backbone with an ester bond. So monoacylglycerols, let's go down here, are designated as 1, 2, or 3, depending on the designation, where this fatty acid is on the number 1, 2, or 3 of carbon. And then we have our diacylglycerols, where we have 2 bound, and tri when all 3 of them are esterified to fatty acids. All the fatty acids are esterified to the glycerol. Okay, and then our most common, or our fatty acid molecule of the most significance, is our triglycerides, or our triacylglycerides, which have the 3 fatty acids on that. And this is 90% of the lipids that we find in our tissues, and it's also the major storage lipid in our bodies. So we do store triglycerides in various tissues throughout our body, but of course the major tissue is our adipose tissue. So let's go a little bit more into detail about one of our types of fats that's really important in our biological processes. And this is a specific type of the glycerol phospholipid that we saw when we talked about the fat types, and specifically a phospholipid. So as we know, these are located in our cell membranes and compose our cell membranes. We don't get many from the diet. Most of that is in the form of triglycerides, but there is a limited amount. And this is a glycerol that's esterified to 2 fatty acids and then a phosphoric acid. And a phosphoric acid has a phosphate group, which of course we know is really important for regulating our biological processes. And then it also has an additional component, which is an inositol, a choline, a serine, or an phenylamine. So we have all these different kinds of phospholipids depending on what that last component is. So the phospholipid that is of a phospholipid that is of biological importance is phosphatidylcholine. And we also know of this as lecithins. And this is a specific type of lecithin, lecithin dipalmitoyl lecithin, is really important in our lung surfactant, which is going to be especially important for our newborn infants, because when they are born premature, or premature infants, I should say, it's this lack of surfactant that's making it difficult for them to breathe on their own. So, so we measure this phosphatidylcholine in order to understand how much help this infant is going to need. Then we also have the phosphatidylethanolamine, phosphatidylserine, which is important for cell signaling, as is phosphatidylinositol and phosphatidylglycerol. So these are all just different kinds of phospholipids. Okay, and then we also have, so again, I'm just kind of going over important fats in our bodies and in our diets. Okay, so we also have our ether phospholipids, including plasmalogens and is it platelet? Yeah, activating factor. So again, there's many types of fats in these categories, but I'm just kind of focusing on the ones that are important and that you'll see clinically. Okay, so these ether phospholipids include plasmalogens, which are complex membrane lipids, um, but the fatty acid at one of the carbons contains just a different group. And these are important in cardiac tissue and in muscle tissue. One of our choline plasmalogens that is important as a biological mediator is our platelet activating factor. And of course, you'll see this commonly in labs clinically because it serves as a mediator of hypersensitivity. It's can measure inflammatory reaction and anaphylactic shock. Okay, so almost all of our metabolism I'm going to save for next week, but this is just going to, I'm just going to introduce a little bit of metabolism here because it's important in our digestion and absorption part of the lecture. So when phospholipids are degraded, it's by an enzymes that are a group of enzymes that are called phospholipases. And the specific phospholipases target different fatty acids on different carbons. So they might target a C1 or a C2 fatty acid. And one of the most important of these that we focus on is phospholipase A2 because it is responsible for the release of arachidonic acid from the C2 position of membrane phospholipids. And then this arachidonate is a substrate for prostaglandin and leukotriene synthesis. So that's going to be important in our conversation about our essential fatty acids and our eicosanoids. So just kind of like, don't worry about it. If this seems a little out of place, just kind of file it away for later and we will come back to it. Okay, so that's really all I'm going to do about classifications of fats because we have so much to talk about with lipids that I want to move on. So here are your practice questions. And now we're going to move on to digestion and absorption of fat and of lipoproteins. So our learning objectives here are to describe digestion of lipids throughout our digestive tract, describe absorption of lipids into the enterocyte and understand how they get into our blood, and think about how maybe defects in these processes could impact our clients clinically. And then also we're going to discuss, because we're talking about digestion and absorption, we are going to take it just one little step further and talk, we know that lipoproteins are important in digestion and absorption, but they're also important for cholesterol and fatty acid transport. So we're going to kind of take that one more step and talk about the transport of the lipoproteins from the liver as well. So first, let's talk about lipid digestion. Now, not much happens in the mouth other than the chewing and kind of mixing this food up, really, but nothing really of clinical significance happens in the mouth. Now, in the stomach, however, we do have enzymes that are going to start to break down the fats. And we know that it's an enzyme that breaks down fats when it's called a lipase. Makes sense, right? We know that enzymes end with ase, lipase. So in the stomach, we have gastric lipase that does hydrolyze a small amount of fat, though much less than we'll see occurs in our small intestine with the help of our pancreas. But it does help start to break down the fat, and actually it can compensate if an individual has deficiencies of other lipases lower in the digestive tract. So some of the breakdown that's going to happen in the stomach is hydrolyzing a triacylglyceride into a diacylglyceride plus a free fatty acid. So we're starting to break off those fatty acids. And then we have our stomachs kind of churning and mixing everything up. And this gastric lipase is most active in the acidic environment. And also gastric lipase is more relevant for, more of an important role for infants because they don't have these enzymes that we'll talk about lower in the digestive tract in as high of quantities. Okay, then the fats would enter, or the lipids would enter the small intestine. And when fat enters the small intestine, it signals CCK, or cholecystokylin, which tells the gallbladder to release bile, and that emulsifies the fat. We know bile, we'll talk more about bile, but we know it kind of acts as a detergent to break up fats and kind of distribute it more throughout the chyme. Okay, so bile and pancreatic secretions are needed to digest and absorb lipid into the jejunum. So this uh, activation or the signaling of CCK and then of bile release is absolutely crucial for lipid digestion. Okay, and so when the fat enters the small intestine, it also the also what is signaled is this pancreatic and intestinal lipases, and these play a really important part in further degrading the triglyceride, or I could have put triacylglyceride into just the glycerol backbone, and free fatty acids, and also monoacylglycerides. Okay, so I think we all know about bile, but let's just do a quick review. So at one end of each bile, there's a side chain of an amino acid. Let's see here. So that's what this R is indicating, this amino acid. And that part is attracted to water. And then at the other end is this sterol group, and that's attracted to fat. So in that way, this bile can really kind of break up fats and emulsify them, not break up fats, but emulsify them within a hydrophilic environment, basically. So before the bile acids, all the fats are kind of clumped together because they're all hydrophobic, or hydrophobic. But then the bile acids come in and kind of break it up with this hydrophilic amino acid part. And then after emulsification, the reason for this is that after emulsification, the enzymes can get at the fat and break up the fat droplets a lot more easily. Okay, so of course, this digestion is taking place in the small intestine, but a crucial part of lipid digestion is these secretions from the pancreas into the small intestine. So the pancreas secretes many lipid enzymes that release fatty acids from a portion of the lipid in the small intestine lumen. So first we have pancreatic lipase that is again going to degrade into monoacylglycerol and free fatty acid. And the pancreas also secretes bicarbonate so that this pancreatic lipase can work optimally. And also colipase that is necessary for lipase binding. And if you're interested in that, you can read more about that in your book. Okay, we also have then phospholipids. Oh, sorry. Phospholipases, please fix that. Phospholipases that are degraded at the second position by phospholipase A2 that we talked about just a few minutes ago. And what that does is release a free fatty acid and a lysophospholipid. So a lysophospholipid is when one of the acyl groups of the phospholipid is removed by hydrolysis. So this will come up, that this lysophospholipid will continue to come up. And then okay, I see, this is just a different structure. So it's also has cholesterol esterase in order to break up these cholesterol ester bonds, and that hydrolyzes free cholesterol in the presence of a lipase, which is secreted by the pancreas. Okay, so here is a nice figure from our book that kind of sums up lipid digestion in the small intestine. So let's just go through it really quick. So we have a lipid droplet or triacylglyceride in the stomach. It encounters the enzyme gastric lipase and then is a diacylglyceride and a free fatty acid. This goes into the small intestine, and think here is secretions from the pancreas, such as all of our lipases and esterases from the pancreas, and also bile acids from the gallbladder, and these encounter this carboxyl lipase and pancreatic lipase with its colipase, and ultimately these bile acids um, emulsify these fat droplets, and these encounter these enzymes so that we're ultimately broken down into free fatty acids, monoacylglycerols, and free cholesterol in these mixed micelles. So we'll talk more about micelles moving forward um, in the small intestine, and that's so that they can be absorbed in the small intestine. And we'll go into that part a little bit more. And so definitely look at this description and walk through it and make sure that you understand what they're talking about, because I think it is a pretty good explanation. Okay, so we're still in the small intestine. Okay, so we know, again, this is just kind of a review of what we just talked about, but we know that lipids kind of clump together because they're hydrophobic, and so they're not very soluble. So we need bile acids that have this hydrophobic and hydrophilic side um, to break up these fats. So bile acids are produced in the liver and converted into bile salts, and these bile salts form micelles, which we need in order to absorb lipids. So what the micelles do is they shuttle products of fat digestion from the lumen to the brush border where they can be absorbed into the enterocyte. So really these fatty acids and other fat products and molecules cannot be absorbed into the enterocyte unless they are part of this micelle. And you can see why a micelle is important, because we have the lipid here, and then we have these bile acids all along the outside, and that's going to help emulsify them so that they can be accessible to these enzymes. And when these fatty acids are in the form of micelles, then they can passively diffuse into the enterocyte. And I do think they talk about a little bit in the book that there may be such a thing as carrier mediated fatty acid transport, but this is still a topic of discussion. Okay, so let's summarize what we just said. So, in the intestinal lumen with pancreatic lipase, we do already have some diacylglycerides from gastric lipase, but in general, for those triglycerides that are left, they're going to be broken down into diacylglycerols and monoacylglycerols, and free glycerol and fatty acids. And so here, so we have our triglycerides, they encounter bile salts and lipase, and then they go to our fatty acids and monoacylglycerides. So these are then incorporated into these mixed micelles, and when they are incorporated into these mixed micelles, they can be absorbed through the brush border. And these micelles can fit in between the microvilli at the brush border. So once they then go into the brush through the brush border, then they start to break up again. Then these micelle kind of starts to come apart with the free fatty acids and monoglycerides, and then we actually put these back together in our enterocyte into a triglyceride, and then these are packaged up with proteins. Hence the name lipoproteins, and all of these are incorporated into chylomicrons that then go to the thoracic duct. Okay, so here's a close-up of the actual absorption of the micelle at the brush border. So in the presence of bile, monoglycerides form into micelles, allowing them to be soluble in the intestine. So here we have our lysophospholipid that we talked about and our bile acids. And then at the brush border, the fatty acids and monoglycerides leave the micelle and are absorbed into the intestinal cell. And then once they're in the intestinal cell, the monoglycerides again reform into triglycerides once they're in the enterocyte. And then they're going to be packaged into the chylomicrons. So we're all kind of talking about the same thing. So this is again kind of like a review of what we've just talked about. So after the micelles are after they're absorbed, the micelles are broken down back into the monoacylglycerides and fatty acids, and then they're reesterified once they're in the enterocyte into triacylglycerides and incorporated into the chylomicrons with the addition of protein. Also, so remember, what we have mostly in our micelles is these monoacylglycerides and fatty acids, but we also have our lysophospholipids, and these are reesterified back into a phospholipid, and the cholesterols are reesterified back into cholesterol esters. So these were also present in the micelles. So once the triacylglycerides and the cholesterol esters and phospholipids are back in the enterocyte, then they're repackaged with the protein into chylomicrons, and these are then transported to the lymph where they enter the blood via the thoracic duct, and then they're delivered to other parts of the body. And chylomicrons are made and secreted only in the small intestine. So keep that in mind, that chylomicrons are a type of lipoprotein, but that is the type of lipoprotein that's only made and secreted by the small intestine. Okay, and here's another graphic that kind of is an overview of what we've talked about. So I think this is a really nice description actually, because they give you numbers and talk you through the whole thing. But basically we have our dietary fat as a triglyceride droplet, as large triglyceride droplets. So they're kind of clinging together. They encounter the bile salts, and we have emulsification so that they're all broken up. Then they have better access to this pancreatic lipase and the small intestine lumen. Then the lipase breaks the lipids down into monoacylglycerides and free fatty acids primarily. We also know there's some cholesterol products in there and so on. Again, they encounter bile salts and are incorporated into these micelles, or also called mixed micelles. The micelles can fit through the enterocytes, through the microvilli of the enterocytes, and then they kind of break back up again into these monoacylglycerides and fatty acids. And then those are reesterified into triglycerides. They're aggregated and coated with, they're aggregated with proteins and coated with lipoproteins they're aggregated with proteins and packaged into chylomicrons, and then they go through the lacteal duct into the blood. So I would encourage you to look through that one and make sure you understand it. And then we also have a YouTube video here that I'm going to let you watch on your own. That's an overview. So now we're going to switch our topic a little bit and focus more on lipoproteins and also apolipoproteins. And we'll talk about what these are and why they're important. So lipoproteins are, as they sound, complexes of lipids and proteins held together by non-covalent bonds. And each type of lipoprotein has a characteristic mass and chemical composition, density, and it has its own role. Oh, sorry. So trying to figure out what I meant there. So the cores of these lipoproteins have cholesterol esters and triacylglycerides, and they also contain phospholipids, free cholesterol, and apolipoproteins. Now, what are apolipoproteins? Apolipoproteins are involved in the assembly and secretion of the lipoprotein. They provide structural integrity to the lipoprotein, and they're really important for activating lipoprotein enzymes. They also serve as a ligand for receptors and membrane proteins, so they're important in cell signaling, and we'll see that moving forward. Okay, so our major lipoproteins that we find in our blood are our chylomicrons that we just talked about, secreted by the small intestine. And then we also have very low density lipoprotein, low density lipoprotein, and high density lipoprotein. So obviously these are biological markers or blood markers that we use frequently in nutrition practice. And these lipoproteins, again, characteristic size, density, amount, and major type of lipid and type of protein. Then the apolipoproteins coat the lipoprotein particles and serve for transport and signaling functions. And the major classes of the types of lipoproteins we see are A, B, C, and E. Okay, so we already touched on one of lipoprotein, which is chylomicrons, which is important for transporting dietary cholesterol and triglycerides to the rest of the body. And the predominant lipids in the chylomicron are triacylglycerides. The next type of lipoprotein we're going to talk about is oh, sorry, not the next type. So before I continue on and talk about how we get to these other types of lipoproteins, I first want to talk about an important enzyme that we're going to see throughout our discussions on lipids, and that's called lipoprotein lipase. So lipase, we know, is an enzyme for lipids, and lipoprotein lipase is a specific type of lipase. So what this lipase does is while this lipoprotein is moving throughout the body, it can lyse off these fatty acids. Yes, they can lyse off these fatty acids from the triglycerides and deliver them to different tissues. So in order to, so you can see that this is a really important enzyme when we're talking about fat transport in our body and distribution in our body. So lipoprotein lipase has to be activated, and it's activated by one of our apolipoproteins. So this is an example of how the apolipoprotein can serve as a signaling molecule. And that apolipoprotein, C2, is found on the surface of endothelial cells in the capillaries. So as our chylomicrons moving throughout our body, it encounters this lipoprotein lipase on endothelial cells, and that's going to start lysing off these fatty acids from the triglycerides that are being transported in the chylomicron. And these free fatty acids are then absorbed into the tissues and utilized for energy, or they're re-esterified into triglycerides so that they can be stored. And then once all the free fatty acids are lysed off, then the glycerol backbone is delivered back to the liver or the kidneys. Okay, so once the chylomicron has been transported throughout the body, and the fatty acids have been removed, then we have what is left as a chylomicron remnant. And because, so think this is our chylomicron, okay? And once all of these have been left off, what really happens is that just by proportion, we have many less triglycerides. So here was the triglyceride in this peach part, and we have a greater proportion of cholesterol. So these are considered cholesterol rich. And the apolipoproteins that are important on these chylomicron remnants are ApoE and a form of ApoB, which is B48. And these apolipoproteins allow recognition by the liver so that they can take up the remnant via a receptor-mediated endocytosis. So the liver sees these apolipoproteins and says, that's what I'm going to absorb, and engulfs it via endocytosis. Okay, so now let's kind of look at a bigger picture. So what we've been talking about this whole time is really this exogenous pathway for dietary fat and cholesterol, okay? So we have dietary fat and cholesterol, it goes into the intestines, it's packaged into chylomicrons, goes into the capillaries, free fatty acids are released by lipoprotein lipase and go to our tissues, the remnant goes back to the liver and is stored in the liver. So here's our dietary cholesterol from our exogenous fat, okay? If someone obviously takes in dietary cholesterol, which of course you don't need to because our body makes it. So next I'm going to focus, we're going to take this a step further, and we're going to talk about this endogenous pathway for cholesterol and fatty acid transport. So once these dietary cholesterol are back into the liver, then we're going to either the liver is going to repackage it as cholesterol in bile acid and excrete it back into our intestine, or it's going to package it for distribution to the rest of the body in the form of VLDL. So I'm just doing an overview here, and then we're going to go into more detail. And so the VLDL then goes back into the capillaries from the liver, and again, we remove free fatty acids via lipoprotein lipase, and then ultimately, it's going to be converted as these fatty acids are lysed off more and more. We're going to get to intermediate density and low density lipoproteins, which are going to go back to the liver and to our other tissues other than the livers, our extrahepatic tissues. And then they're going to be picked up by HDL, which ultimately takes them back to the liver. So we'll talk in more detail, but this is a really nice kind of overview figure of just fat and cholesterol transport throughout the body, both exogenously and endogenously. Okay, so let's move on to our next lipoprotein. We talked about chylomicrons, which were exogenous from exogenous pathway. Now let's talk about the lipoproteins and our endogenous pathway. So let's start with VLDLs, very low density lipoproteins. So first thing we should know is that when we consume excess fat than we need, or even if we consume excess dietary carb, which can be converted to fat, then those fats are going to be converted to triacylglycerides in the liver. Okay, so we saw in our last picture here, okay, that we're getting some of the triglycerides and cholesterol kind of left over from our digestion and absorption. But then we're also making triglycerides from excess fat and excess carb in our liver. So now we have all this cholesterol and triglycerides in our liver. What's your liver going to do? It's going to package these into very low density lipoproteins and release them into circulation for delivery of these fatty acids and cholesterol into the tissue. And they're primarily where they can either be stored or used for energy. And that's going to be primarily in our muscle and adipose tissues. And of course, mostly in our adipose tissues. Okay, so then as the VLDLs are going throughout our body, it's the same enzyme that's going to help release the fatty acids from these, our lipoprotein lipase. So this is a really important enzyme for fat distribution and transport. Same thing as the chylomicron. It's going to use this lipoprotein lipase to lyse off these fatty acids for the other tissues. Okay, so as this VLDL is going throughout the body and it encounters the lipoprotein lipase, so it's losing fatty acids and it encounters a loss of one of our apolipoproteins, apolipoprotein C, then what we're left with is that the VLDL is converted to an intermediate density lipoprotein. So it starts off as a VLDL, then we're going to lose one of our apolipoprotein C, and we're also going to lose our fatty acids because of lipoprotein lipase, and we're left with IDL. So we don't hear about IDLs that much. It's kind of like an intermediate step, so I won't focus on it too much. But basically, once the IDLs occur, the same thing is going to happen. We're going to lyse off more fatty acids, and it's going to become LDL. And the LDL can be taken up directly into the liver. I just don't want to repeat myself too much because I don't want to confuse you. Okay, so we have the VLDL, we're lysing things off, it becomes IDL, we're lysing more things off, it becomes LDL, and then that LDL can actually be endocytized by the liver. So it has apolipoproteins on it that the liver recognizes, and so it lands on an LDL receptor, and the liver endocytizes the LDL. And okay, so LDL is then the primary plasma carrier of cholesterol for delivery to all tissues. So this LDL is what's going to kind of go throughout the body and kind of deliver all of this cholesterol into tissues. And we know that that can have an arthrogenic impact. So LDL exclusively has the apolipoprotein ApoB100. And it's this ApoB100 that is going to allow the LDLs to be to land on the LDL receptor of the liver and be endocytized by the liver. Okay, so this is just talking about some of the, this slide is just referring to some of the cholesterol aspects of the lipoproteins, but I think we'll focus a little bit more on that next week. So if you don't fully understand this slide, again, kind of file it away for later, and we'll talk more about it next week when we talk about cholesterol specifically transport. Then we have our HDLs, and we all think of HDLs as kind of our good cholesterol. And this HDL is primarily protein rich, and it's a disc shaped particle. And because it's so protein rich, that's why it's called a high density lipoprotein. And because so the main function of this HDL is not to deliver cholesterol to the periphery, but rather to remove cholesterol from the periphery to the liver, where hopefully it can either be repackaged or excreted. Um, so HDL is synthesized in the liver and the small intestine. Its most predominant apolipoprotein is ApoA1. Um, and again, we'll talk more about LCAT next week when we talk about cholesterol transport specifically. But free cholesterol that are in IGLs that are moving throughout our body can then be esterified through this enzyme called LCAT that's associated with HDL. And HDL requires the interaction of this ApoA1 that we find on HDL on the surface of HDL. Okay, so what HDL does is it removes excess cholesterol from the periphery and facilitates its transfer back to the liver, where it's then converted into bile. And think about it, when it's converted into bile, then it's going to be excreted into the small intestine in order to help with our digestion of lipids. And some of that will probably be reabsorbed in the end, but some of that then is going to be excreted. So this is kind of our only way to excrete cholesterol from our bodies. And then once HDLs have gone throughout the body and collected this cholesterol from the periphery, so they're endocytized by the liver, and then again, it will repackage, the liver will repackage this cholesterol into bile. So, and then again, this process of the HDLs collecting this cholesterol from the periphery and taking it back to the liver is called reverse cholesterol transport. Okay, so here is a big overview of our exogenous and endogenous lipid transport systems, okay, including cholesterol. And again, we'll do more detail on this when we talk about metabolism, but let's just kind of break down this figure, okay? So first we have fat from our diet, right? Goes into our stomach, goes through our intestines, into our enterocyte, into chylomicrons, and then these into our capillaries. And we know that the chylomicrons deliver fatty acids to the periphery, and what's left is a chylomicron remnant, okay? And that these are cholesterol rich. So these chylomicron remnants then go back through a receptor back to the liver, and we have our cholesterol in our liver. So this cholesterol can be repackaged into bile salts, and then this goes back into our digestive process. But we can also have other outcomes for this cholesterol from the chylomicron remnant. So this chylomicron remnant, along with triglycerides that are in the liver from excess carb and excess fat, are going to be packaged into VLDL lipoproteins. The VLDL goes back into the capillaries, and when free fatty acids are lysed off, then it becomes IGL and then LDL. And LDL is what transports cholesterol, the most of the cholesterol into our peripheral tissues. This LDL cholesterol then goes back to the liver and is endocytized via LDL receptor transport. And then again, it can go back into bile salts or it can be repackaged into VLDL. However, the cholesterol can also be, the liver can also synthesize HDL. And HDL is going to go to our peripheries and it's going to pick up our cholesterol and bring it back to the liver where it can go into bile salts or be packaged right into VLDL again. So that's kind of our overview of how cholesterol and lipids are transported throughout the body. So, as we can imagine, if HDL is picking up cholesterol from the body and delivering it to the liver, and part of it will be excreted, then it has an anti-atherogenic role. So, because it's picking up cholesterol, that's going to impede the oxidation of the cholesterol that's contained in the LDL molecule. And we know that this LDL molecule or oxidation is what is kind of the initiating factor of plaque buildup in the arteries, causing arthrogenesis. So this figure is just kind of a way you can conceptualize the differences in the chemical makeup of the lipoproteins. So in chylomicrons, it's the most triglycerides, and that makes sense, right? Because most of our diet is composed of triglycerides. So then it's going to go throughout the body and come back, drop off a lot of these triglycerides and come back and repackage in the liver. And you can see at that point the VLDL has many less triglycerides and many more cholesterols and phospholipids. And then it's going to go throughout the body and lose some of these triglycerides, lyse off these fatty acids, and it's going to become kind of a higher proportion of these cholesterols to phospholipids. At that point, it's going to again encounter more of this lipoprotein lipase and lose even more of these triglycerides. So we have an even greater proportion of cholesterol in these lipoproteins. So, and then that low density lipoprotein is going to go back to the liver. However, we also have these HDLs that are synthesized by the liver, and you can see these are high density because they have a much higher proportion of proteins and nearly no triglycerides and much less cholesterol. All right, so now I want to talk about, you've kind of been hearing me throw around this apolipoproteins, A, B, C, whatever. Let's focus on what these mean. So again, sometimes macronutrients can be difficult to explain because it's not really a linear process, but really all of this stuff is happening kind of at the same time and is so highly interconnected. So we have to try to break it apart so we can understand it, but it makes it difficult to explain everything at the same time. So you've heard me talk about the apolipoproteins. We know they're important for signaling. Let's look at the specific types of apolipoproteins, what lipoproteins they're associated with, and why they're important. So first we have apolipoprotein A, and as we said, the specific type A1 is a major component of HDL. And this apolipoprotein makes cholesterol efflux from the periphery to the HDL effective. So it's really this apolipoprotein that's the first step of reverse cholesterol transport. So why this apolipoprotein is this ApoA1 is so important is that it serves as a cofactor. So it's traveling along an HDL, right? ApoA1 is traveling along an HDL, and it serves as a cofactor when the HDL encounters this enzyme called LCAT, okay? So, and what LCAT does is enzymatically releases the cholesterol so that it can be incorporated into the HDL. So what it's really doing is this ApoA1 is facilitating this reverse cholesterol transport. So we know that that when we have more ApoA1, when we see more ApoA1 in the body, that's associated with decreased cardiovascular disease risk. And that really makes sense, right? Because if we have more ApoA1, we have more reverse cholesterol transport, we have less plaque development. Then we also have apolipoprotein B, which exists in two important forms. So first we have ApoB48 that, as I said, is present on the chylomicrons and their remnants, and plays an essential role in the intestinal absorption of fats. We also have ApoB100, which is a major ligand for the LDL receptor. We have apolipoprotein C, which, as I said, is important in chylomicrons and VLDLs, and also a necessary cofactor for lipoprotein lipase activation. So think about that. These chylomicrons, let's say chylomicrons, but it's also VLDLs. We know one's just the exogenous pathway, one's the endogenous pathway, right? So let's say in this instance, the chylomicron is traveling around with this apolipoprotein C on it, and this apolipoprotein C is a cofactor for this lipoprotein lipase that's going to be encountered in the capillaries, which, when it works, when it has this cofactor, then it can lyse off these free fatty acids for distribution to the other tissues. And then we have ApoE, which is synthesized in the liver from VLDL, and its role is recognized by the LDL receptor in the liver. So cholesterol will not be taken up into any cell. Also, cholesterol will not be taken up into any cell unless it's present with ApoE. So this cholesterol must have this ApoE on it in order to be taken up into any cell. So some individuals may have a genetic defect in producing ApoE, but really in those individuals we haven't seen a defect in lipid metabolism. So we kind of know why it works, but we don't know that it's exclusively responsible for this cholesterol, this cholesterol enzymatic reaction. So again, we know its role, but we don't really fully understand it. Okay, so here is a nice overview of everything we've talked about so far. So again, when you're studying, go through these figures and make sure you understand just what is happening here. Okay, so this is kind of an overview. We have a chylomicron that's packaged in the small intestine from our dietary intake, and it's highly with triglycerides. It's heavy on the triglycerides, and it has this ApoB48 on it. Then it's going to go through be delivered into the capillaries, and here we see that ApoC and ApoE are transferred from HDL, that's also in the blood, onto the chylomicron. So then this chylomicron has all three of these apolipoproteins and goes into the capillaries. We know that then some of this, one of these apolipoproteins is necessary for activating this lipoprotein lipase, and free fatty acids go to our peripheral tissues. The leftover glycerol goes to the liver, and the chylomicron remnant is delivered back to the liver. At this point, the ApoAC is then laced off again and goes back to the HDL. So then we have our chylomicron remnant, which still has our ApoB and ApoE, and it's these apolipoproteins that are necessary for this cholesterol-rich remnant to bind to the receptors on the liver where it's endositized. So this is all still kind of our exogenous pathway that we talked about. Okay, so actually we see that even though we know that how important lipoproteins are in the distribution of fats throughout our body, that defects in lipoprotein metabolism that actually cause clinical outcomes are kind of rare. However, we do see that patients with certain clinical outcomes such as diabetes, hypothyroidism, kidney disease do have abnormal lipoprotein metabolism as kind of a secondary effect. Now, we're going to kind of start tying in some of our metabolism from carbohydrates here, because we also know that lipoprotein lipase is regulated by our hormone insulin. So that lipoprotein lipase deficiency could lead to hyperlipoproteinemia as a secondary outcome of diabetes. Okay, so here is kind of an overview of both the endogenous and the exogenous pathways. So let's go through it. Okay, so hopefully you're like, hey, I'm sick of hearing this repeated right now. But I think it's important to walk through all these multiple times just so you get it. Okay, so we all get it. So we have our dietary fat and cholesterol, goes to a chylomicron, goes into our capillaries. Triglycerides are hydrolyzed by lipoprotein lipase. Glycerol, or the fatty acids, are delivered to the peripheral tissues where they can be stored or oxidized. That's what we're going to talk a lot about next week. And then the glycerol backbone can be returned to the liver for glucose synthesis. Remember when we talked about gluconeogenesis and that one of the substrates was glycerol from triglycerides? Okay, so that's kind of how this ties in. And so that's our, and then once these fatty acids are lysed off, the chylomicron remnant then goes back to the liver. Then with this cholesterol that's gone back and the triglycerides that's gone back, along with the fats and cholesterols that are synthesized in the liver, these are packaged up into VLDLs. Same thing, they go into the capillaries, and we go through this whole process again where we're lysing off fatty acids with lipoprotein lipase. Then the VLDL is converted into IDL. That can go back to the liver, or it can further be broken down by lipoprotein lipase and go to LDL. Okay, the LDL can then go back to the liver via an LDL receptor and endocytosis, or it can go onto an LDL receptor on our peripheral tissues and deliver the cholesterol to these peripheral tissues. So we know that LDL is the main transporter of cholesterol to our peripheral tissues. However, then our HDL that has been synthesized by the liver, can come along and pick up some of this cholesterol. And this cholesterol, we're going to see next week, can be used for its own synthesis of biological compounds, or it can take the cholesterol back to the liver. And that cholesterol can then be excreted in the feces or be repackaged into bile acids. Okay, and here are your practice questions for this second part of the lecture. So again, I know this is kind of introducing hopefully some familiar material, but also some new material. And I'm always available for questions, as is your TA Chelsea, and it's great to talk to your classmates too. So because this lecture is on a week where I know you're working on your first exam, your first take-home exam, we're not going to have an article discussion or this week. So for our discussion board, we'll really be talking about some of the concepts that I discussed in this lecture. So I will talk to you all next week, and I hope you have a lovely week.